Oh, Stephan, it's awfully quiet around here on midterm break. Whew. Gives us a chance to catch up a little bit.
No, it's been a wild ride in Bio 125 so far. I mean, a lot of information has been exchanged between faculty and students, from phylogenies to protein synthesis. I can't wait what the second half of the course holds. How do you think it's gone so far?
I think it's a fantastic class. I feel really good when I'm in there listening, and they have such great questions, a lot of energy. Cheers to the Bio 125 class. I really did think when you were talking about introns, I think that maybe you missed one part.
So the introns, the junk DNA actually within a gene that separates the exons.
Intervening sequences that get spliced out. And at that point you said, really it doesn't matter a lot what the DNA sequence is, except where it's cut.
The junctions, yeah.
Yeah, the junctions are really important because that's where it's cut out. But then, what about the interior? It doesn't matter. Then I did think that it might matter in one circumstance. Maybe we can go to the board and I'll show you what I mean.
Sure. Let's take care of this question here.
All right. So yeah, this has to do with introns and how the sequences in introns might matter. So if you've got this section of DNA here,
Drawing it double-stranded, great.
Double-stranded. And say it's a long piece, and some of it is exon, that's the coding sequence, and then there's an intron, that's the part that will be, is the intervening sequence that will be spliced out. And so on this DNA comes, I'll draw this big cloud here. This is RNA polymerase. Reading along, transcribing the message. This all makes sense to me, but my question is, what happens if this triplet is a stop codon? I think this breaks the rule that the intron sequence doesn't matter, because this RNA polymerase comes along and hits a stop codon. So I think this would be a negative mutation that would be thrown out by natural selection, because it would stop the reading. If there's another exon out here, it would never get there. This would be a really negative mutation, even though it's a sequence inside an intron. So why doesn't that cause all kinds of trouble?
Well, Mark, I think the trouble is arising is that you're making a classic mistake in confusing transcription and translation.
The words even sound the same.
I know, but we have to keep them straight. Remember, transcription in a eukaryotic cell in the nucleus, translation out there in the cytoplasm. Even more important, who's doing the transcribing and who's doing the translating. The RNA polymerase is doing the transcription, correct? The ribosome is doing the translating.
Oh.
So if I could take your diagram a little further, okay, yes, let's say there is a stop codon, a UAG sequence right here that codes for a stop codon. I'm going to go with my purple pen here there. So we'll have a transcript of this region in the nucleus. So here is our messenger RNA. What happens to messenger RNA before it is allowed to leave the nucleus to go to the cytoplasm?
That's the splicing.
That's the splicing. So this intron region will be cut, and you said the stop codon was in the intron region?
Right.
Oh, I'm sorry, it is gone. These two exon regions will be spliced together to make the mature messenger RNA. And this sequence now will be translated by the ribosome. So the ribosome will attach, and codon by codon, it will convert this sequence of nucleotides in the mature messenger RNA into an amino acid sequence. So your stop codon in the DNA was a little bit of a misrepresentation. I mean, strictly speaking, a stop codon has to be in RNA. RNA polymerase does transcription, not translation. RNA polymerase does not even recognize codons, stop codons, can start codons. It's the ribosome that's responsible for recognizing the AUG start, all the triplets, and then the final stop codon. Nucleus, cytoplasm, transcription, translation.
So at this stage, RNA polymerase is not doing anything to understand the message. It's just DNA base pairs. Stop, start, methionine, whatever, it doesn't know. The translation mechanism is down here.
Right. All RNA polymerase can do is to make an RNA copy of the DNA, exons, introns, whatever. It's just letters that it's converting into a messenger RNA sequence, and then it's the ribosome that is decoding that.
Makes total sense. So I will happily talk about how intron sequences are irrelevant for evolution, except at the cutting site.
Very good, very good.
Excellent. Thank you.
There you go. You know, it looks like the faculty is out there getting ready for the faculty broomball tournament. We better head out there or we're going to be late. Come on, let's
That's
get
great. I'm
upstairs.
going to break some legs.
All right, let's go.